We're continuing the seminar series with two PhD plan talks. They're going to be 20 minutes talks with probably around 10 minutes for questions, just so we have enough time. First, we have Rocío Fuerte, who's going to be talking about ungulate effects on Mediterranean shrubs. Thank you very much.
Thank you. So, good afternoon, everyone. I'm Rocío Fernández, and I'm doing my PhD here at the EBD with José María Fedriani and Pedro Garrote. As you can see, my thesis is about the interaction between overabundant ungulate populations and a common shrub species of the Doñana National Park. To introduce a bit about myself, I'm from Córdoba when I completed my biology degree, and I started collaborating with the plant and ecology department. Later, I moved to Seville to complete my master program in biodiversity and conservation ecology biology in the University Pablo de Olavide. That's when I first arrived to the EBD to do my master thesis. That's with the same group I'm working now, and that master thesis is what led to my PhD. So, in this master project, we obtained results about the nature of the interaction between overabundant ungulates and the plant I'm currently studying, and we saw how often does the ungulate visit the plant, and how the consumption of the different reproductive structures affect the plant survival. Right now, these results are under review by a journal, and we are waiting for its publication. So, after that, in February of 2024, I started my PhD thesis here. So, as I have only been here for a year, my talk is going to be about what have I done in my past year, but mostly what we intend to do in the future years. So, my thesis is included in the theoretical framework of interspecific interactions. Species interactions are often classified as mutualistic or antagonistic. However, it's important to note that the same interspecific interaction can have both mutualistic and antagonistic components, which can fluctuate depending on factors changing in the ecosystems. That means that an interaction is existing in an antagonistic mutualistic continuum that fluctuates depending on ecological factors giving place to an antagonistic mutualism continuum. So, to know how the interspecific interaction work in our ecosystems, it's important to know how these ecological factors vary and how they influence them, such as the density of populations, its distribution, and the phenotypes of the species involved. Plant-frugivore interactions are widely considered as mutualism, as they are basically a trade-off of food for the animal for long-distance seed dispersal for the plant. This process of seed dispersal is crucial for the establishment of animal dispersed plants, as many of them have evolved to be dispersed by frugivores. Nevertheless, the animals involved in this interaction can also act as predators, as they can destroy the seeds that they ingest of these plants, reducing the survival rates of this species. So, frugivores can become both seed disperser or seed predators, and creating a mutualist-antagonist duality, really important in nature. Endosoochory studies have mainly focused on fleshy fruits, but dry fruits, such as the Cistacia 1, can also be dispersed by animals, such as fish, rodent, birds, and ungulates. Ungulates are key factors in the configuration of plant populations, as they are present worldwide and in a great variety of ecosystems, such as the Mediterranean ecosystem. High population of ungulates are present worldwide and in protected areas, such as the Doñana territory, have suffered a remarkable increase. Ungulates, specifically the red deer, which is the main character of my thesis, is present in all habitats of Doñana, but they show a preference for some sites over others. The red deer show long daily displacement that determine the population's density and distribution, creating differences on their population's density in the different areas of Doñana territory. So we have to make an effort to understand how the red deer affect the environment in order to get a better knowledge about the population's dynamic. In my previous results of my master thesis, we know that the red deer can act both as a mutualist or an antagonist, and that the net sign and magnitude of this interaction can vary depending on ecological factors such as the ungulate density and the ontogenetic stage of the individuals involved. So in my thesis, we studied the interaction between the shrub Halimium halimifolium and some overabundant ungulate populations in the Doñana National Park, focusing on the red deer. I'm sure you mostly know this plant, but Halimium halimifolium is a woody shrub from the Cistaceae family, which is the principal component of the shrubland ecosystem that we know in Doñana as Monte Blanco. As you can see, it has yellow flowers, and the fruit is a dry capsule which can contain up to 50 seeds per fruit. Their seeds are white and soft when they are immature and dark and hard-coated when they are fully mature. Its fruiting and flowering period occurs between March and August approximately, although it's a plant that shows a high asynchrony on its phenology. So the main objectives of my thesis would be to separate the mutualistic and the antagonistic components of the interaction between Halimium halimifolium and the red deer, and therefore obtaining a better knowledge about how the sign and the magnitude of this interaction fluctuates in space and time. So basically we want to evaluate the role of the red deer on Halimium, differentiating on the one hand on its paper on its role as a seed disperser, and on the other hand as a consumer of flowers, immature fruits, immature seeds and leaves. So we have chosen these two species because surprisingly, although they are two of the most common and iconic species of the Doñana National Park, the interaction between these two has been little studied and overlooked until now. And the effect between the interaction between these two species can also cause great effects on the rest of the ecosystem. So for this purpose, we have developed 4 different chapters. The first one would be the evaluation of the red deer as a seed predator or a seed disperser on Halimium halimifolium, testing the evolution in the consumption of the different reproductive structures of the plant. The second chapter would be about the study of the retention times of the seeds in the animals' interiors, focusing on the red deer. Going to the third chapter, we evaluate the seed survival rate and germination rates in laboratory experiments, and the fourth chapter will also be about germination experiments, but this time in the Doñana substrate. And the fifth and last chapter would be to integrate the results gotten in the past chapter in order to get a global perspective through simulation models. So first of all, the first chapter of my thesis is aimed to evaluate the role of the red deer as a seed disperser or a seed predator on Halimium halimifolium by evaluating the consumption and the herbivory of the different reproductive parts along its phenology. So, as I have mentioned before, an interspecific interaction can be mutualistic or antagonistic depending on factors such as the ontogenetic stage of the plants. So, in this specific interaction pair, we evaluate the benefits and the cost of the interaction depending on the maturation of the different reproductive parts of the Halimium halimifolium. So, to make this clear, because it's the key concept of this chapter, the red deer would be considered as a predator if the consumption of the plant occurs mainly when the plant presents flowers and immature fruits. And on the other hand, the red deer will be considered as a mutualist and a disperser when it consumes the plant when it mostly shows mature seeds and mature fruits, as the red deer will facilitate the long-distance seed dispersal via endosoochory in the Doñana territory, assuming that the seeds will survive the pass through the digestive tract. So, this is the most advanced chapter till now. After a field season we developed from May to September of 2024. To do so, we selected 3 study sites that host 3 different deer populations density, being from higher to lower, Matas Gordas, Reserva, and Sotos. So, on each study site, we differentiated to different habitats depending on its distance to the marsh. La Vera, which is the closest habitat to the marsh, would host a higher density of deer population than the interior shrubland habitat. So, in each habitat, we differentiated 4 plots between the period of February and March of 2024, creating plots of 6 per 6 meter approximately. So we had open and closed plots. the open plots were defined using iron rods in the corners, whereas we built closed plots using a 2 meters height ungulate exclusion nets. So we described 4 types of plots. One plot that is open the whole time of the experiment, one plot that is closed the whole time of the experiment, and then the other 2 are exchanged with the maturation of the fruit. It means when a high percentage of the fruit of the plants of our plots are mature, and an open plot was closed and a closed plot was open. So we do this mechanism in order to evaluate the effect of the presence of the red deer, separating the mutualist and the antagonist phases of the interaction. So we have a closed plot when the plant presents mostly flowers and immature fruits to focus the experimental design on the antagonistic and predator role of the red deer. Whereas we have a closed plot that only in the period when the plants contain mostly mature fruit. So we focus the experiment on the time where it will occur the mutualistic part and the dispersal of the mature seeds. So this experimental design is made to evaluate the effects of the red deer in 4 different scenarios in order to get to be more precise when comparing the mutualistic and the antagonistic role of the red deer between the 4 of them. So on each plot we selected 8 plants at the beginning of the field season, that was when the plant contained mainly floral buds and open flowers. So on each plant we selected inflorescences that were labeled and monitored the rest of the field season. So on each inflorescence we counted the number of floral buds, open flowers, immature and mature fruits, and aborted and lost fruits. So at the end of September we obtained the weekly register about the evolution of the herbivory and the development of the reproductive structures of the plant from the flowering period till the end of the maturation of the seeds. So after a first field season and some preliminary analysis and some personal observation we had on the field, we can infer that the consumption of Halimium halimifolium is predominant when the plants contain mainly flowers and immature fruits, so there would establish a more antagonistic and predatory role of the red deer on Halimium. Nevertheless, we still have a second season of field work ahead of us that will take place from May to September of 2025, and we still have to make a more exhaustive design of the data analysis. So in my master thesis, we developed camera trapping monitoring, concluding, as you can see in the bar chart, that the red deer is the main visitor of the Halimium halimifolium individual. So in my PhD, also in the past field work, we also developed camera trapping monitoring, but this time differentiating in 3 moments of the phenology of the plant. When the plants host mostly flowers, mostly immature fruits, and mostly mature fruits. So we set the cameras in mesh towers, as you can see in the picture of my colleague, pointing to the open plots and being activated when movement is detected. So after this monitoring, we obtain graphic evidences about how the frequency of the visits of the ungulate species to our plants, but this time differentiating between the 3 different phenological stages, and also learning if they are making use or not of the plant. We are already analyzing these photos, and we are taking note about the species that we see in the photos, the number of individuals, if they are making use or not of the plant, if they are inside or outside the plot, the date and the time of the photo, and some other personal interesting observations we consider, such as the sex of the individuals when it's possible. Moreover, in our herbivory monitoring, we found a high quantity or frequency of invertebrate animal, most of them Lepidoptera caterpillar, so that were damaging or consuming vegetative and reproductive parts of our plant. So we consider that it's an important factor to take into account when studying the herbivores that influence the reproductive success of Halimium. So when we made the herbivory monitoring last year, we also take note about the species, the name of the species when it was possible, and the number of individuals. And once we finished the field season and the monitoring season, we went once a month to our plots, first to make revisions about how the state and the evolution of the phenological process of our plant, and also to get a register also of the species and the number of individuals of these caterpillars, in order to obtain the temporal evolution through the whole year. We described 7 species of caterpillars, being the most abundant ones: Saturnia pagonia, Silobaster loti, and Lasiocampa trifoli. So here we have the temporal evolution through the whole year, and we can see that the maximum peak of caterpillars was in June, when the flowering period was ending and was starting the maturation of the fruits period. So as in general terms, we can see that the amount of caterpillars is remarkably decreased once the summer finishes. Now, moving on to the second chapter, is about the study of the seed retention times in the digestive tract of the red deer. Basically, we want to know how long does it take for the seeds to be expelled from the animal's body via their feces. So, knowing this useful information, together with data about the daily displacements of the red deer in the territory, we can obtain an estimate of the maximum distance to which the seeds can be dispersed via endozoochory by the red deer in the Doñana territory. So, for this purpose, we designed an experiment with captive red deer in the wildlife park El Osquillo in Cuenca that will take place in August of 2025. To do this experiment, first we will give and feed the animals with mature seeds directly collected from the plants, with a food that facilitates and ensures the total ingestion of all the seeds provided. After that, the rest of the days of the experiment, more or less 10 days, we would collect the feces from the red deer, registering the moment of collection. So when we develop the fecal analysis, we would know how many days does it take for the seeds to be expelled, and also how many of the seed provided would survive to the path through the digestive tract and the fecal feces samples. So here we have a sample of mature seeds directly collected from the plant. Then this was a trial that we made in a zoological park near Seville, but because of logistical reasons we had to change locations. And here we have a fecal sample, and it's processing with a mortar under the loop. Now, in the third chapter, we want to evaluate the seed survival rate and the germination of the seeds that had been ingested by the red deer and exposed via their feces. So we hope to use the seeds found in the feces of the previous chapter to create the experimental group, and then we have to collect some mature seeds directly from the fruits of the plant to create the control group. So we set the seeds in petri dishes with distilled water in germination chambers in the laboratory, and the days after we make daily revisions to compare how many control seeds and how many ungulate ingested seeds manage to germinate. So with previous results about of my master thesis, we expect that the ungulate ingested seeds germination rates and survival rates decreased as they are damaged with the mechanical and the chemical digestion via chewing or the intestinal fluids. However, we still a small group of the ungulate ingested seeds got to survive, so it gives the red deer an important role as a seed disperser of the plant. So it opens up the possibility of a mutualistic component in the interaction between these two species that is of great importance when maintaining the connectivity between populations of Halimium distant in Doñana's territory. And lastly, the last 2 chapters of my thesis are less defined, but we have the following proposals. For the first chapter, we also intend to develop germination experiments, but this time using Doñana's substrate, so we could germinate them in the laboratory or directly in the field. And we expect to create exclusion fences around the possible appearing seedlings in order to prevent them from trampling or from the consumption of other herbivores. And with this experiment, we could obtain more realistic results about how the plant and its germination behaves in the field, taking into account other environmental factors such as the Doñana soil's properties that would affect the plant. And finally, in the last chapter, we could integrate the results from the previous chapters and create future scenarios in order to create a more global perspective about the interaction between these two species. So we hope to design simulation models that integrate the results from the previous chapters, the red deer densities, and the moment of the phenology of the plant when the interaction takes place, to separate the mutualistic and antagonistic components of the interaction and see how it behaves across space and time. So, with all the results gathered in my thesis, we hope to deepen our knowledge in the interaction between these two important and overabundant species in Doñana, and to get a higher knowledge about how the sign and the magnitude of the antagonism-mutualism continuums varies across time and how it affects to the rest of the environment. So, thanks to all of you for your time and attention, and to my group for trusting me in this project, and now I'm happy to answer all of your questions.
Thank you. There's plenty of time for questions.
Thank you. Very interesting, very nice presentation. But I have some issues about your idea about predation and dispersal.
Yeah.
Because in the first chapter, you consider that all the fruits that are consuming, they are disperser. No.
No, when we make the herbivory monitoring, we intend to differentiate, because some of the seeds are not eaten; they fall or they.
Yes. But in fact, if the red deer eat the fruit, then a part of these seeds will be dispersed, but another part will be consumed. Because you have seen how the rate of germination is very low after passage in the red deer. And my question is, if you could sample from the feces of the red deers, you collect excrements, and then you try to identify your seeds in these excrements, and then you can try to see if they are able to germinate or not.
Yeah, that's what I did in my master thesis. That I don't think I have mentioned it, but the first chapter of the first part was to the collection of feces in the field, and then the analysis, the fecal analysis in the laboratory. So with the seeds we found in the feces, that it was a really high percentage. Then we set them to germinate. So these seeds came from a collection of feces of Doñana.
Okay,
so
thanks. And another question is, for your field germination experiment in the field, in the chapter, this one, which kind of seeds you will see in the field after a red deer passage, or only seeds, natural seeds from the plant, or the two kinds of seeds after passage or not?
Yeah, we should still have to define this, but it depends really, because theoretically the seeds that came from the ungulate ingestions came from the captive red deer.
From the captive.
Yes, so it depends on how many of them we obtain, because we have to use them in the lab experiment that I think is more defined. And if there are lots, maybe we could use them in the field, or in the field maybe only focusing on mature seeds that haven't been ingested. But it's true that this experiment wouldn't be so reliable if we didn't use the ingested one to our objective. So, yeah.
Okay, Thank you very much.
Thank you.
Thank you, Rocío, for your very nice talk. So I was curious about the natural history of this interaction, because unlike other shrub species, Mediterranean shrub species such as myrtus, pistacia, discus, etc., fruits of Cystaceae do not seem adapt to endozoochory. They just mature, open, and the seeds are delivered. This is the first time I hear about endozoochory in this family. So I was wondering if this interaction between the deers and this plant is something eventual. How common is this plant in the diet of these animals or in other animals? I don't know. What do you know about this?
Well, as what I have read is that the dry fruits that are adapted to endozoochory have not really developed structures to this endozoochory. They just produce a lot of seeds, small seeds that could get to survive the digestive tract. Historically, I don't know how to tell you, but the interaction between these two, as the phototrapping results show that they interact a lot. I don't know if I'm explaining, but because of the abundance of both species, it should be a really common, but we had to extrapolate the small results of the small group of ungulate ingested seeds that get to germinate to the whole territory of Doñana and the whole population of Halimium and of the red deer. So although it's from the total of ingested seeds, supposedly a small part only gets to germinate, but if we know how much do they interact, the effect on the ecosystem and the population survival would be really high.
It would be interesting to see this interaction in other regions, for example, just for the sake of comparison, maybe outside Doñana, but I'm talking for the future, not for your thesis.
No, there are studies not with halimifolium, but in the center of Spain with other cystus individuals.
Okay, that's very nice.
Yeah.
And just a minor question, different question. I am very interested in the recruitment. So I was wondering if these animals, the deer, also predate on the saplings, the baby plants of the halimifolium.
Yeah, it's one thing that we have to take into account that we haven't, but it's not only that the red deer predates the plant when it's an adult, but we also have to consider the seedlings that they might ingest. But yeah, it would be interesting to know how really occurs and maybe to simulate the results about the interactions to take this into account, but we haven't.
Thank you very much.
Thank you.
Hi, Rocío. Great talk. Based on your preliminary data, I'm wondering if you know if sex or body size of the deers impact the germination rates of the seeds.
Well, I haven't read anything about this, but in the captive red deer experiment in Cuenca, I haven't told this, but the experiment is designed to have for individuals, maybe one female, one male, and two middle-aged red deer. So here we could also compare both ages and sex of the individuals. So we still have to prove this.
Okay, thank you very much for the presentation. It's a lot of fun. But you talk a lot about deer, but you don't explain the kind of information you are going to have for the deer. Because I was looking at chapter 5, it seems that you will need information about the movement of the deer. Are you going to get information? Do you have, what kind of information do you have
Yeah, I still have not the information. Well, we know that more or less they make displacements or 3 or 4 kilometers per day, more or less. But it also depends on the moment of the day. But we still have to do a bibliography search or maybe collaborating with some registers of Doñana.
Yeah, also because I think that this information, probably there is plenty of information from other populations, but I don't know for Doñana, that is a very unique place, and also that relates also to the title, because you talk about overabundant deer. I mean, you think that the situation for the deer and this plant is exceptional because of the abundance of the deer, or you think that could be a reflection of what is going everywhere else in the distribution?
I think it would be extrapolated to the rest of the peninsula, because it's, as I have read, the amount and the frequency of these ungulates have increased not only in Doñana but in most protected areas and in more environments of the peninsula. So I studied this specific interaction comparison the zones of Doñana, but it would be interesting to see what happens in other parts of the peninsula or other zones, and maybe with other cistus species that are look like Halimium. Maybe.
Thank you.
Okay, I say we thank Rocío again, and we'll move on to the next talk.
You all for coming here. Thank you. Yeah, my name is Francisco Javier Borrallo Vázquez, and for the next 20 minutes I'm going to present you all my thesis called Evolutionary Implications of Host and Parasite Interactions, Insights from a Computational Approach, which is supervised by Miguel Fortuna here from this house. So, as everybody knows here, mutualistic interactions are actually drivers of diversity. They promote new niches, they create and then maintain those niches along perturbations. As the other side of the coin, antagonistic interactions, here focusing on parasitisms, actually have the same effect. They promote new niches, they push hosts outside the boundaries of the and make them diversificate a long time. This study has been present since the last century, in the early days of the last century, but has a really good boom at the mid and the 50s with, for example, the Red Queen hypothesis. The problem with this kind of studies is that following coevolution along hundreds or maybe thousands of generations, it's a very taxing and it requires lots of effort. So that's how we are going to do this computational approach to try to overcome this barrier. So I'm going to start by posing the main objective for my thesis. We want to understand the mechanics behind coevolution of host parasites, because we think that by understanding the mechanics, we can then guess the dynamics and help them guide those coevolutionary trajectories towards more desirable ends. We are going to understand them deeply. As I said, to know all this information, to have all this data, you will require lots of effort. If we imagine a normal microbiology lab, it will need lots of material, lots of personnel, and overall lots of time. And since I don't have that time, because I'm doing a thesis only, I'm using this computational approach to try to overcome these barriers. So, this is my artificial life lab. This is my computational approach. It's based on AVIDA. AVIDA is a system that can replicate evolution in a digital interface, in our computers. It stands in a balance between the mathematical models and the simulations, and it draws from the generality, from the capacity of mathematical models to abstract the dynamics of the system and then tell us very clear, and between also laboratory experiments and field research, because it goes a step beyond and gets more closer to the randomness and the realism of the natural world. So, how is AVIDA able to do evolution in silico, since it's only a computer program? Well, within AVIDA, there's such things called digital organisms. AVIDA really can only do, in the base form, can only use evolution. It can show you evolution in a computational way. But in our lab, we have extended that functionality to be able to replicate also coevolution, focusing in the coevolution between a bacteriophage, a virus bacteriophage, and then his bacterial hosts. It does so because AVIDA has this thing called a digital organisms. In basic form, a digital organisms is just a self-replicating computer program that will evolve and will mutate within the user-defined computational environment, within the memory of our computers. I'm going to explain you very briefly what are the components of a digital organism so that you will understand how can we achieve evolution in silico. So, like the genome of a bacteria, a digital organism is composed of a series of instructions that it must execute sequentially, and by doing so, it will gain a phenotype. The phenotype we call this is the mathematical functions these digital organisms can have, for example, addition and subtraction. You can think of this as a bacteria that is able to metabolize glucose and then fructose, for example. It also provides them a fitness that we can measure very specifically with the replication rate of these organisms. Then we have the genotype, phenotype, and fitness, the three components for evolution. We need then the spark to lead this evolution engine, that these are mutations. These digital organisms can mutate inside the environment. It can change and add some different instructions to their genome. So by the addition of mutations over time, digital organisms can really evolve new mathematical functions, for example, multiplication. In this case, this new organism can metabolize, for example, amino acids, so it can extend to new niches. And also, they will replicate faster. So these organisms, these new organisms, will spread over population because it has greater fitness than the ones that came before. This is actually evolution in our computers. So what we have done also is to be able to have a new kind of organisms that will be the role of parasites. Okay, first of all, these organisms will be living inside the memory of our computers, but you can toggle with them and you can configure the experiments in a way that will replicate those in a normal microbiology lab. Knowing so, we have configured these digital organisms to interact with each other in the same that it replicates a ChemStat. We have created a digital ChemStat. For those that are not familiarized with this, a ChemStat is a very simple device that is composed of a bioreactor that will host the bacteria, the organisms, and they will live here. Then we constantly pump them some growth medium. We will replenish the resources of this dynamic environment, and at the same time we are going to take out some contents of the bioreactor as waste. It will be resources, bacteria, and wastes. This simple contraction has three major advantages that people like Monod postulated in the 50s, and those are that we can control populations at the steady state. By the amount of nutrients we get into the system, we can control actually the abundance of the populations at a very constant steady state. We can control the growth rate by knowing how much of we dilute our original ChemStat, and by controlling the growth rate, we can also infer in things such as genetic drift. The higher the dilution rate, the higher will be the genetic drift inside our ChemStat. And the most important thing maybe is that all the populations that we will here will be mathematically tractable. There will be models that really align with what the populations are doing here. As an example, this is a graph of our digital organisms evolving with our digital ChemStat, and you can see here how well the models, the mathematical models, adapt to the same organisms, but evolving under three different dilution rates. So, with this, these are the components that I'm going to use to resolve my thesis, and the four questions we did to ourselves to try to unravel the dynamics of host-parasite interactions are these four, and they are going to be the chapters of my thesis. It's if parasites' virulence on their hosts decreases over time, if parasites increase the epistatic interactions of their hosts, if parasites increase the information content stored within those host genomes, and finally, if coevolution is more repeatable than evolution. These four questions are very broad, are very theoretical, but they will give us a lot of insights on the dynamics of host and parasites coevolution in different perspectives. What I'm going to explain now, instead of each chapter, I'm going to explain the experimental design we have developed to try to tackle these four questions at the same time. So, all of our experiments will start with a digital host as a parasite and a digital organism as a parasite and a digital host here. The key here is that for the parasite to infect the host, they need to share at least one part of their phenotypes. They will need to have a common mathematical function, so they can interact with each other. In this interaction, in this infection, the benefit that the parasite will win by stalling the replication rate of the host is what we call the virulence, that we will signal here by the shape and the intensity of this arrow. So over time, we expect that this digital host will evolve a new phenotype, a resistant phenotype, that will allow this parasite, will allow the new host to escape the parasite, but the parasite will counteract this effect. It will pursue their host over time. This here is what we call evolution, coevolution. So at the end of thousands of generations, if they coexist, we expect to have a lot of strains of parasites interacting differently with lots of different strains of hosts. We are looking here at the phenotypical traits, we are looking at phenotypical things, but we can also have information about the genotypes. So, for example, for any individual within this evolution, we can know their genome, actual genome, over long periods of time. If we focus, for example, in any individual at the end of the experiment, we can actually know what mutations took place from the ancestors to the end of the experiment, when did those mutations happen, what was the effect of the mutations, and the actual instructions that mutated. So we can actually go back and see the whole evolutionary trajectory of, for example, the most successful individuals at the end of the experiment. We can go beyond, we can go to the population level, and we can also save the complete genomes for both hosts and parasites at several different time points. We're going to save like we are fetching it, and then explore the whole genome diversity of these populations. And then finally, we are going to do this, but saving the population at different time points. Closer or further from the original outcome, we are going to see what are these alternative outcomes, and we are going to compare them between them to see what the effects of the distances from the actual end time. This is only one experiment we're going to do, but notice that we have 10 different host ancestors with 10 different genomes and 5 different parasitic ancestors. Each unique pair will be replicated 20 times, and since we have 50 pairs, we are going to have 1,000 experiments. 1,000 experiments during thousands of generations, so we aim to have a very thoroughly perspective on our initial conditions. So now I'm going to explain what part of this experimental design goes to tackle the specific chapters. So starting with the chapter 1, if virulence decreases over time. So as I said before, we are going to see the evolution of virulence between the different phenotypes of hosts and parasites interacting in a coevolutionary scenario. But thanks to our computational approach, we can bend the rules of this scenario without changing anything else, and we can shut down the phenotypical matching, the rule that makes that for a parasite to infect a host, it needs to share the phenotype. So by doing this, we are actually swapping from a coevolutionary treatment, we call it here treatment, to an evolution treatment. Why? Because all the infections will be possible no matter the phenotype hosts and parasites will have. That means they will evolve independently. They will not interact with each other in any way, but the infection. So the only selective pressure that will appear here will be this pressure for both hosts and parasites to replicate as fast as possible. That is here noted in the intensity on the green color. But we can go also beyond, we can go a step beyond and have the ground truth. So we can shut down mutagenesis, we can have no mutations over time, and then see how virulence and the population of the ancestors behave over time during the same time. So what do we expect to see here with the virulence to be able to construct a hypothesis from the ground? Well, for the ecological treatment, we expect to have a quick and very steady virulence decrease until they reach the virulence optimum for both populations. For the evolutionary scenario, we don't expect virulence to decrease much, and that's because parasites have two ways of increasing their own replication rate, that is evolving and also maintaining a high virulence. And then for the coevolution treatment, that is very interesting in this treatment, we expect something in between. Why is that? Because when we have a lot of different parasites coexistence with a lot of different hosts, we can have an arrangement of the architecture interactions. So, for example, interactions can arrange in a very specific way, where specialists only interact with a specialist, and under this scenario we expect virulence to be very high as there is a fierce competition. But there can be interactions that can arrange in a generalist way, in a very nested way, where virulence can rearrange instead of the interactions. So maybe lower levels of virulence will be selected as they are the best strategy for some parasites. This is only one of the hypotheses we are going to test. As you may understand, we have a lot of data, so we are going to test this hypothesis thoroughly. Now, we are going to go to the genotypical information, to the genomes, and what we are going to do here to know if collision with parasites enhances epistasis is we are going to go and take the most successful individuals at the end of each experiment, and we are going to trace the genome back to the ancestors. As I said before, we can know in each step of the coevolution what was the change in the genome and what was its effects. So we are going to have the whole evolutionary trajectory. Now, we are interested in the if parasites have an effect enhancing the epistasis of the hosts. So for those that are not very common with epistasis, it's simply we are under epistasis when the effects of one mutation, the fitness effects of one mutations, are not independent and depend on the presence of other mutation. So to know epistasis along the whole coevolutionary trajectory, we need to compute every single point mutant possible within each step, for example, the single point mutant 1 and the single point mutant 2, and then assuming that we are under no epistasis, the fitness effect of the double mutant will be simply the addition of the fitness effect of these single mutants, making thus the double expected. But since we are using this computational approach that is so useful, we are going to actually characterize and compute all the double mutants that are possible within each step of the genome. We are going to actually know what is the epistatic, the real fitness landscape, and not only that, we are going to see the develop on all the trajectories, on all the evolutionary trajectories from the ancestor to the actual outcome. So we're going to see if parasites enhance epistasis. So what we are guessing here, that's if we start here, this is the epistatic effect distribution of our ancestors. Some might be negative, some might be positive, antagonistic or synergistic between them. I represented it here very simply in these boxes. The size of these boxes represents the even distribution of beneficial and deleterious effects, antagonistic or synergistic between them. Over time, if parasites enhance epistasis, what we aim to see is that the distribution of epistatic effects will become something like this. As you can see, the size of the boxes has changed, the amount, the value of the fitness change is less. Why is that? Because the coevolution with parasites might push the host, the distribution of fitness effects to the host to something like this, while the deleterious antagonistic effects get enhanced relative to the others. So in this scenario, our hosts will have less fitness lost than we expect. The fitness valleys will be less deep. To test this, we actually need to know if this is a property of the genome or is caused by the presence of the parasites. So if you remember, we have the evolutionary treatment that we are going to compare with this one. Now, this is for the individual level, genomes for the individual level. We are going to go to the population level, and we're going to save all of these genomes to answer the question if parasites increase the information content on their host genomes. To explain this, I will need a bit of information theory, but hold with me, it's very easy to understand, as many here will be common with this. To do this, what we are going to do is calculate the entropy of these genomes each time step. And why are we going to calculate the entropy? Hold the thought here. Entropy is also called uncertainty and follows this formula. It's a very common formula for ecologists, as they do when they calculate the Shannon diversity index. They calculate the entropy of an ecosystem. And what we are going to do here is calculate the entropy for each position of each genome for both hosts and parasites. And then average for the whole population in different time points. This, as you can understand, it's a very taxing thing to do, even if you are going into a computational approach. But why are we doing this? Because information stored in a genome is manifested via conserved sequences. If we are going to have a conserved sequences, it's a sequence that we are going to be certain it will repeat over time. In other words, the entropy of that genome will decrease. So, by having the entropy of the populations at all levels, we are going to know if they are storing information at what pace. Actually, we are going to make an amount of information index, and we are going to track it for hosts and parasites, mostly for hosts, over time. So we expect to see that if parasites increase the information content, the fall of entropy for those genomes under the coevolution treatment will be faster and will be deeper than the fall of entropy for the evolutionary treatments. This means that there will be a fixation rate that will be faster in both terms of amounts of, and growth of those conserved sequences for the coevolution treatment than the evolutionary treatment for the same genomes of ancestors. And then finally, we are going to see if coevolution is more repeatable, because if something is certain to will happen, then by definition it also becomes predictable. What we are going to do here to see this is we are going to have our experiment with the original outcome, and we are going to save the population and replay the tape of coevolution at different time points. For example, we are going to have the T1 time point, it's going to be very close to the origin, and the populations coexisting in this time point will be replicated and let them co-evolve over time for each replicate. So we are going to have for the first pair 20 replicates of these alternative evolutionary outcomes. We are going to do this also for the second and for the third time, and many other different time points. What we expect to see here is that the similarity between these alternative outcomes will be greater at the end of the experiment than at the start of the experiment. And why is that? Because at the start of the experiment, the genomes still have a lot of potential. They can diversify and they can take many different paths. And that potential gets lost the more we advance in evolution or coevolution. For coevolution, it's very simple. We are going to check, for example, the network architecture. But for evolution, we are going to look into other things such as the fitness, as we can have very close measures of fitness. And then, in a quick summary, we're going to track virulence along diversification. We are going to also track the genomes of the successful individuals and recover the evolutionary path to see the evolution of epistasis, track the genomes to see how they accumulate information, and then compare the replay coevolution and compare the repeatability of those outcomes between themselves. This is more or less my current progress. I'm more confident in the first two quests. And then finally, some knowledge, my group, the Bioinformatics and Computational Group, we are a small group, but Miguel and Fortuna, that's my group's leader and test supervisor, and Raúl Ortega is the magic hands that make this possible, and also the Life Hub computation, the Life Hub connection and the BCP connection. Thank you all.
Thank you very much, Francisco. Questions? Anyone?
Okay, thank you for the talk. In one moment of your talk, you have justified how you pretend to shift off the coevolution.
Yeah, when shutting down the phenotypical matching, right? This was at the start.
Can you justify it again, because I have
not
understood
the idea is there's a rule to interactions. This rule is modeled after the phenotypical matching of a virus entering the bacteria through the membranes receptors by matching the phenotypes of the tail receptors with the virus and the bacteria. This is the same. What we are going to do here is, without changing anything else, we are going to allow all infections to be possible. So there will be no actual benefit to the fitness of the host or the fitness of the parasite in changing their phenotype, changing the mathematical function they can do, changing what they can take from the environment. Here, without changing anything else, the shift, there will be no benefit to change, to diversify. So actually, they will evolve independently. The only pressure, the selective pressure that there will be in this environment, it will be to replicate as fast as possible. Hosts will try to replicate as fast as possible to try to push parasites into extinction.
But the matching is the only thing that is codified in the genes of...
The phenotypical matching.
I mean, even if the infection is random?
No, here it's not random. They need to share part of their phenotype. They need to do a mathematical... Yeah, it happens. In this part, it happens no matter your phenotype. All infections are allowed. At random, yeah.
But anyway, the genes of the parasite affect the fitness of the host.
Yeah, that's the only selective pressure we have, that is to replicate as fast as possible.
And vice versa, the genes of the host affect the genes of the. Then it could be coevolution anyway.
I mean, they have an effect in each other, but they will the diversification they will have, it will be independent.
No.
They won't. They can diversify, but they don't. They are not diversifying. They are only replicating as fast as possible.
I mean that the matching of the infection is only one part of the possible pathways to evolve because of the genes of the other part, no?
Yeah.
I mean, no. Need to take it for
it's a very
what I
interesting.
say, but you can test it.
Yeah,
anyway.
we should be able to test that.
You
But like,
say, well, infection at random, but
the reciprocity is broken in the evolutionary scenario. So it doesn't matter if there is no phenotypic matching. The parasite can always infect the host. That's why you don't have coevolution, because the evolution of the parasites is not driven by the evolution of the hosts.
But one thing is to infect,
Mm-hmm.
and another thing is what happened then. Or this is not something that.
The parasite has a negative effect on the host, but the host can never escape from being attacked.
But the thing is, you are attacked or not. Not what
No,
happens
you
when
can always, parasites will always be successful in infecting their hosts in the evolutionary scenario.
The
But
evolution is only about
in the coevolutionary scenario, you can have total resistance to all parasites, or partial resistance. A host can be partially resistant to some parasites. But that reciprocity is broken in the evolutionary scenario.
The outcome of an interaction is simply being infected or not being
Yeah.
in the model.
The outcome, it's going to depend on how much the parasite or how the negative effect that the parasite causes on the host, which is the virulence. That's the only thing that is going to change in both the scenarios.
mm-hmm.
But the virulence is after the infection.
No, the virulence is actually the percentage of how many resources will get stolen by the parasite. They will coexist. So I get the idea what you're talking about, but here that's why I say there will be a pressure that will increase. That will be the effect of the infection of the parasite. That will be, they will try to replicate as fast as possible. This is also maybe if there can be other kind of effects of the infection model that will not depend in the phenotype of the parasites, but this is the simplification we have used to be able to kind of build up the different scenarios, so we can know the evolutionary behavior of these organisms, and then we'll up to the coevolutionary scenario.
So thank you for the talk. I'm very nice and clever design. So are you entering some other kinds of stochasticity into the picture, or I mean, when you look at the repeatability of coevolution versus evolution, I guess that depends on the level of stochastic processes, randomness, and so on. So I guess so. I didn't understand whether that is kept constant in both scenarios, or maybe you play with different kind of levels.
We try to control the stochasticity of our model the best we can. That's why we use the digital chemstat, because we can control mathematically everything. It's one of the chapters I need to work most on to check this actual similarity. It's very easy to understand in the evolution, but we try to maintain at least the same condition of stochasticity between all the replicates. In the evolutionary treatment is more complex because there will be other forces in play. So I'm going to actually check more that, but we are going to try to be as as constant as we can with those non-controlled factors. Technically, we can even know how many resources are there in the model at any time, in real time. So I hope we can get maybe a comprehensible degree of stochasticity between these outcomes to see not only for the coevolution interaction to see if they replicate at the same time, if the genomes are more or less the same at the end of the experiment, we can know many things. So I don't know.
But I guess the responses are contingent on the level of randomness in a way, or maybe the initial conditions as well. So yeah. I guess I'm trying to make up my mind on whether, the outcomes are very dependent on that specific factor, or they can be general.
Outcomes are depending on the initial factor of the ancestors, for example, the replication rate of the hosts. The initial replication rate of the host will be very important for the evolution of virulence, and there for the evolution of the wall trajectory. Mm-hmm. So we kind of know, we are going to, when we have the data, we are going to feed it to the models to kind of understand in different time points, not only in the first and the last one, to see what are the things that actually matter for the wall coevolution. So yeah, we kind of know those kind of things. Yeah.
I think you mean stochasticity between the replicates of the same experiment using the same pair of ancestors. The stochasticity comes from the way mutations take place. So in each replicate, mutations are going to happen in a different way between replicates. So you don't get the same outcome at the end. You don't know. That's the stochasticity that we incorporate in the system. The fate of mutations. In each replicate, you start from the same ancestor, the same genomes, but mutations are going to take place in different ways in each replicate. Yeah. So you don't know how much repeatable the final outcome are going to be because of that stochasticity that you introduce since the beginning of the experiment.
And the other thing is that this is kind of reminiscent of this Richard Lenski's experimental evolution study on E. coli, yeah. In which, they resurrected groups of bacteria that kind of were extinct and competed them with groups of bacteria that were successful. So are you planning on doing something like that? That I think can be cool, maybe because you can play with, say, some kind of bacteria that they became extinct in this timeline. Mm-hmm. And then just you can, I don't know, is there room to play with these kind of things to see also it's along the lines of the repeatability of evolution. I guess it fits with that kind of aim as well. Yeah,
we could. It will be very interesting overall for the evolutionary scenario where we can check bacteria that have, bacteria or hosts or parasites that have been extinct and then compare them to the actual outcome and see if they become extinct in other scenarios, in other replicates. So it will be very interesting. We'll see if we can fit into this, so we can take out the data and we can track extinctions and then take data from that. So yeah, I'll think about it if we can put some of Richard Lenski. I need to read about a lot of the last Lenski's jobs and works to be able to understand everything here. This is the most the chapter that I control the list.
Thank you.
Thank you again, Francisco, and for both Rocío's talk as well today. And we'll not see you next week because of Semana Santa, but have a good Easter.